Good day everyone, my name is Leila Nefdt and I'm a master's student in the Biological Sciences Department at the University of Cape Town. I'll be focusing my talk on epifaunal community patterns within the Agulhas ecoregion in South Africa. We recognize that there's a lack of knowledge in understanding our deep sea systems, especially in terms of understanding patterns and processes around the South African offshore environment. In saying so, we intend to grow our knowledge base of these offshore deep sea areas, particularly along the continental shelf and edge. This is where my project comes in. It forms part of a larger multidisciplinary project called the Deep Secrets, which falls under the African Coelacanth Ecosystem Program. This project aims to develop offshore research capacity and knowledge in support of improving marine management of the outer shelf, shelf edge and upper slope ecosystems of the Agulhas ecoregion. Ecoregion meaning areas of relatively homogeneous species composition that are clearly distinct from adjacent systems. And by surveying these areas, we can then gain an inside perspective on the current community composition and determine the main environmental drivers of the epifaunal communities found along our continental shelf. In turn, this will provide us with vital information for identifying biologically important areas for conservation managers and scientists alike. The general aim of my study is to refine and improve the marine ecosystem classification and map, contributing towards South Africa's emerging marine spatial planning processes. And by using the newly collected biological data in conjunction with physical variables, we will then be able to determine the biogeographic and biodiversity patterns across the Agulhas ecoregion. My study focuses on the Agulhas ecoregion, which spans the area running along the continental shelf edge of southern Africa, from the tip of the Agulhas Bank in the west to offshore of the Kei River mouth in the east. What is interesting about the shelf edge and slope is that its depth varies right across the shelf. The Agulhas ecoregion has a unique geographical position which lies between a cooler Benguela current and a warmer Agulhas current which expands across three different ocean systems, namely the Atlantic, the Indian and the Southern Ocean. A total of 29 stations surveyed were selected for analysis. These stations had a sample depth ranging from 150 to 700 meters. The survey took place on the Deep Secrets Cruise during September-October in 2016. In this project, the most effective technique for deep sea sampling was to use image analysis. By using underwater imagery taken by the SkiMonkey III benthic camera system, specifically a towed camera survey, was used to quantify in situ benthic biodiversity. More specifically, I looked at epifaunal organisms in my project. So when it came to processing the images, I used the software called Transect Measure from CGIS, and this software was used to define the area processed within each image by making use of the three known laser positions and using a series of calibration images captured prior to the survey. Once each image was calibrated within the software, I would then overlay a 20 by 20 dot grid over each image, and all epifauna that were visible underneath each dot, they were counted for and identified as far as possible. I quantified it in a standardized area looking at abundance per meter square. And in total, I processed about 855 images. So these are just a couple of examples of the epifaunal taxa recorded within my imagery. I recorded about 163 of them, and the most commonly occurring morphospecies found along this region were octocoddles, echinoderms, spider crabs, gastropods, ascidians, and bryozoans. In order to understand and make better sense of the habitat types within this region, I visually categorized the substratum type found within each sample, being each image, ending up with 4 different categories, that being sand, gravel, rock, and mixed substrata. When it came to analyzing the data, I used the multivariate analysis approach using the software Primer version 6 with the Permanova add-on package. I used this to compare biodiversity between different depth zones, substratum types, and oceanographic conditions. Using association-based analyses, I forthwith transformed the abundance dataset to downweigh the abundance species, And using a Bray-Curtis similarity, I created a resemblance matrix to produce a cluster dendrogram and an MDS plot, multidimensional scaling plot, and these were used to graphically represent the relationships between the sites. I then ran a simple analysis to determine the characteristic or distinguishing species that contributed towards the grouping of these sites to form communities. And lastly, I ran a CAP analysis, a canonical analysis of principal coordinates. This analysis was used to assist in the categorization of the substratum types per image. With the environmental data, I ran a Draftman's plot to test for correlation between the variables. And using the distal M analysis, I produced a DBRTA plot to graphically represent the relationship between the biological data and environmental variables. Looking at the cluster dendrogram, we can distinguish that there were 9 epifaunal community groups, A to I, with several outliers at an 18% similarity cutoff, as you can see with the dashed line. Each of these groups holding sites with greater similarity with one another. This is the multidimensional scaling plot. It shows us how closely related the sites are within the 9 community groups. And on the next couple of slides, I'll briefly display a few examples of the community groups with their respective depth range, substratum type, and top 3 characteristic species. Group C was a great representation of mixed substrata samples along the shelf edge and slope with characteristic species of hydrocoral, bryozoans, and various ascidians. Group I was another great representation of mixed substrata samples, but on the outer shelf, with characteristic species of cutcorals, sea fans, and bryozoans. Group D was a great representation of gravel samples along the shelf edge and slope, with characteristic species of polychaetes, cereanthids, and cutcorals. And Group H was a great representation of sand samples along the shelf edge and slope with characteristic species of brittle soils, cereanthids, and polychaetes. Looking at the CAP plot, it shows that there is a distinction between the 4 substratum types with slight overlap. Substratum does seem to have an influence on the distribution of these epifaunal taxa. It would make sense, as you would expect to see a different community of fauna inhabiting sand versus rock. However, we decided not to use substratum type in the distal M analysis because we have used the fauna to guide the classification of the substratum types and using that substratum classification in the CAP analysis to force separation among the data cloud along the substratum axis. A distance-based redundancy analysis was run on the resemblance matrix using species abundance with the variables trawling intensity, depth, longitude, current speed, bottom temperature, slope, and distance to shelf break. All 7 of these variables were significant using marginal tests, and overall, all 7 variables contributed towards the distribution of these epifaunal species. Using the best test, we received an R-square value of 0.169, which is quite low. Even though trawling intensity, depth, longitude, and current speed came out on top of the list in terms of having higher variation, these variables explain very little, or should I say just over 11% of the total variation of the observed faunal patterns. In terms of spatial positioning, these community groups do not separate out well. There's an east to west separation within the community groups. As you can see, they literally cross the entire region. Keeping in mind that this is the very first time that we are looking at this region, this first-time study has shown that the Agulhas ecoregion is quite a complex system, presenting a gradient of sand to mixed substrate to rock, perhaps creating patchiness across the shelf edge. It is a mosaic of habitat types along the shelf edge. 9 community types have been identified along this narrow band, with a substantial overlap in species composition between the community groups. Based on what we observed from the CAP analysis, there is a strong possibility that epifaunal patterns are driven by substratum type. But of the measured variables, trawling intensity, depth, longitude, and current speed seem to have a slightly greater influence on the distribution of these communities. However, we appear to be missing the key physical driver contributing the most. If there are any suggestions as to what else we may be missing, please let me know. Thank you for listening, and thank you for allowing me the opportunity to share my work with you all today.